All right, I have a pop quiz. What are the other IRTs called? I'm doing the IRT3, Integrated Research Thrust 3. What are the other IRTs called? I'm just joking. Don't worry, you don't have to answer. But hopefully in this talk, you'll be able to see that we are really integrating these other two IRTs that you've heard so much about the past yesterday and today into this IRT3. So yeah, so I'm covering the center's really accomplishments in this IRT3, and it's in the tissue mechanics in 4D. So I co-lead this IRT with my fabulous colleagues, Chris Chen at BU and Paul Jamie here at Penn. Okay, so you saw this slide, I think, with Vivek yesterday, and so again, when we think about these IRTs, IRT1 is that recursive dialogue with the cells and the matrix, understanding how mechanics influences the cell behavior in that respect. And then the IRT2 is the the nucleus and nuclear memory. And so again, understanding chromatin changes, epigenetic, transcriptional changes, how that will feed back again to the cell's interaction with the matrix. And then ultimately, IRT3 is looking at those cells assembling into tissues and the influence of mechanical cues on that tissue organization as well as, and then tissue and organ function. So really it's looking across many scales, length scales, time scales of the assembly of those tissues and the influence of mechanical forces. So our goal again and our aim in this IRT is to understand the mechanobiology at that tissue level. And we've spent quite a bit of time development of these platforms. So I'm really going to highlight a lot of our platforms or devices that have been developed over the years to address some key questions. Before that, again, I don't have to motivate this group, but why do we look at this? And so here, as you can see, in development of the Drosophila embryo, we see forces are being applied, cells are assembling, and this is a highly orchestrated event, highly integrated event in development. And then you also see there on the right that you have at this single cell level, these focal adhesion dynamics that's controlling cell morphology, cytoskeleton organization. And so again, all of this really kind of comes together. And again, as you begin to form these tissues, cells are really not only experiencing forces, but they're also generating them. So when you think about the blood vessel, that shear stress, that regulates permeability in the endothelium, that ultimately will then affect vessel tone. And then that can ultimately have an impact there, cardiac, in terms of cardiac output. So the organization of cells into more specialized multicellular structures really impacts how cells transmit and experiences those forces. So again, overarching, okay, focus of the IRT3 is again studying mechanics at the multicellular level, tissue level, development of those platforms. And so we spent a lot of time developing these microfluidic and micro tissue approaches to really capture mechanical influences and really also begin to capture some of that structural complexity that you see in native tissues as well as disease states, disease tissues. And then we're applying also theoretical computational models to those systems. So key questions that we've been addressing or looking at overall is how do tissues distinguish and respond to different stimuli, mechanical stimuli? How can we understand, engineer cell organization in response to those mechanical forces? Can we begin to understand the mechanobiology or further understand mechanobiology of cell-cell interactions? How do cells communicate with with neighboring cells in a coordinated way in the behavior and different cross-kingdom systems, plant and animal systems? And then ultimately, how can we look at then mechano-intelligence, okay, at this multicellular level? And ultimately, how does that feed back into nuclear changes and then back to the tissue and remodeling of these tissues? So I'm just going to highlight really, again, these accomplishments really in development of these tissue platforms and some theory that's associated with that. And so we've looked at various tissue structures, and I'm going to start with perfused ducts. So we've got quite a bit of accomplishments in these areas. And this is very relevant, right, to both plant and animal systems. And so I'm going to describe a lot of the work here within the animal systems. So early work with Christian looked at this perfusion system of blood vessel on a chip. So the endothelial cells are lining that vessel, and is able to demonstrate, again, this barrier function, and was able to identify a novel sensor, this Notch1 receptor that drives that, or restores barrier function under different influences. And this ultimately will form this complex, right, with this cadherin, adherent junctions there in the blood vessel. And so Christian teamed up with Becky Wells and formed also another system to investigate bile duct, right? And so this is important. That bile duct barrier function is important in certain liver diseases. So there, those vessels are lined with cholangiocytes. And again, you have to have basically a super confluent layer of cells to maintain this barrier function. And you also have to achieve this polarity there, apical basal polarity in that vessel to have this protective role, really. And that was seen when they passed bile acid through there. So the glycolix was identified as providing that protective barrier. And so again, very neat systems there. And there's other groups that have listed some other names at UC Merced who are actually developing even more complex systems where you can now add the mural cells into slightly different types of devices so that you can look at geometries of these vasculature, as well as this kind of development or remodeling of these vessels. And so those systems, right, were developed even further. So again, Becky and Chris looked at, then let's create a vascularized bile duct on a chip. And so in this case, some of the advances here was one using disease cholangiocytes, so patients who've had some biliary disease, and now incorporating fibroblasts in this intervening layer. I don't have the thing. Intervening layer between the vessels, the blood vessel and the bile duct vessel. And looking at, again, if there's migration by also putting in immune cells. And so they were able to see then that, yes, the immune cells would migrate there between the blood vessel and that bile duct. And this is in response to, and they can even provoke that even further by adding certain cytokines, and then noted chemokines were secreted. This system is also very useful, again, if you're looking at additional branching of those ducts. And so this really recapitulates kind of the stem cell development or this purulary gland development in these systems. So it's a good system in terms of developing, again, further complexity, getting closer to the disease states and understanding how, again, the influences of these vascularization can affect the bile duct. Some computational models, and again, Vivek Shenoy and others looked at here what the effect is here in this this barrier function, understanding then the competition, right, between some of the signaling molecules that go on between ROA and ROC. And so really those endothelial junctions, there's a strong cadherin interaction or tension that goes on. And the accompanying signaling is important to analyze. And so what they found, there's an optimal ratio of that ROA and ROC1 needed in order to maintain a tight junction. So if that's off balance in any way, you get a loss of the or weakened junctions in these models. So again, these types of discoveries are important for understanding or potentially developing some sort of therapeutics in this area, looking at the Rowe family here with these tight junctions. And also a very interesting perfused duct system here. And this is Dan Hu's group looking at looking at then implantation on a chip. And so what this is important again in pregnancies where you've got the embryo needs to be implanted into this endometrium. And so you need to have then or show what they tried to show here in this on a chip model is that those specialized fetal extravillous tropoblasts need to infiltrate or migrate to the maternal blood vessel. And so again, you had these two ducts. Again, you had or chamber of those trophoblasts, and then you had the blood vessels. And it was important to have a few things here. Where you need to have close proximity of that chamber of the close proximity of that blood vessel with those trophoblasts in order for that migration to occur. They also used primary cells, human cells here to investigate these things. And then also when they put these uterine natural killer cells into these blood vessels, again, you also got this substantial increase in invasion of those tropoblasts. And again, so 70% of the lymphocytes that are present in that maternal tissue are these natural killer cells, and they play a significant role in that implantation. All right, and so again, barrier function is disrupted is what they found here in these cases. And again, it helps to further understand what happens here in pregnancy, and then also when pregnancy is not successful. Okay, moving a little bit to some of our other accomplishments with some of the more sheet-like networks, and then some building on some of those complexities. So Chris Chin's group was able to develop these pillars, micropillar structures. So these micropillar platforms have actually are being used quite a bit throughout the scientific community, so not only in our center, but other labs. And so what's nice about here, again, in this work is that he was able to grow those iPS-derived cardiomyocytes, create a tissue that was functionally contracting tissue. And it could create then a model of cardiac infarction by, again, applying this laser that would create a wound. And what was neat in this study is that, again, he utilized about 10% of the population was of fibroblasts, amongst those cardiomyocytes. What they found after the wound creation during healing is that those fibroblasts really grew into large numbers, shown there on the bottom panel. And what remained, the cardiomyocytes that remained, unexpectedly increased contractility in this case to compensate for that loss of tissue and overgrowth of fibrosis. So again, these are interesting models to be able to start to get closer to what you see in cardiac and these cardiac infarctions. And here it's a little hard to see there, I see, but this is an on-chip, a cardiac pump on a chip. And so again, increasing more complexity, what they did was Chris's group was looking at growing those tissues now on this 3D printed scaffold platform that allows for contraction of the tissue in a unidirectional manner. And so took some redesigning of that scaffold to get it such that it only would deflect in a kind of, this one direction. Because once that contraction started, the force could be quite large. And so initial attempts there showed that it kind of collapsed and didn't reflect or deflect back. But they were able to figure that out, that design there, get that to work well. And then what they were able to show, and I think the video hopefully will work, with the addition of valves, again, you can't see it in that device, they were able to get unidirectional flow in that device. So as this thing is, you see it's contracting and moving, you have a supply of the flow, right? A supply channel mimicking kind of the venous flow. And then you have an output channel, right? That arterial flow. And those things are happening then at different times. So that supply channel goes in, and then you have the output channel. So what that really imitates and what they were able to demonstrate is that you are able to get this pressure gradient volume loop, PV loop generated in this device. Other devices that have been made looking again at tissue, some of this even tissue morphogenesis, I would say, in devices where you can constrain or create these either isotropic or anisotropic types of mechanical changes in the tissue. And so what you can see here is that, again, if you constrain those two ends, these are collagen gels with cells embedded, these are stem cells. And if you constrain the two ends, you get contraction there along that principal stressor axis. They're shown on the left. Cells respond, and they will differentiate into tenocytes in this case. If it's isotropic, fully constrained on all sides there, you don't see this response. You also don't get differentiation of the cells. And so again, you can organize tissue and begin by the influence of mechanical stimuli. Looking again at some other more challenging types of cultures would be your organoids. So here, again, if you're developing organoids, say out of stem cells, iPS cells, in this case, looking at lung alveospheres, getting improved homogeneity out of those organoids is important. And so trying to move away from matrigel systems here is important in this, and this is Burdick's, some of Burdick's work. And so developing improved materials like hyaluronic acid with very well-defined ligands, laminin in this case, and also mechanical properties. So there was an optimal storage modulus or modulus that was able to get a more homogeneous organoid in this case. So a fair amount of work has been done there. And then moving on to also Dan Hu's group has been able to also address an important point of organoid development in the lab in these cultures is the loss of viability in the core of these tissues or growing tissues. And so he's been able to kind of redesign and simply create these what's called an octopus system by these inserts that go right into your 96 well plate or culture dishes. And it allows for, again, the growth of a fair amount or fairly large growth of these organoids that are fully viable throughout the kind of core of these organoids. And he's even improved some of these techniques by allowing for vascularization. So again, you can also feed in these endothelial cells into some of these ports, and it allows for full vascularization of these organoids. So again, these types of devices are being looked at for a number of diseases or development of lung tissues, for example, or cancer models. And then another interesting and always neat to look at, right, is this blinking eye. So this is, again, an ocular surface that was created or recreated here on a chip system. Hydrogel, that hydrogel is an electromechanical kind of actuator, again, that goes across that surface of the eye. So you've got the cornea cells and you've got the conjunctiva cells, epithelial cells that are present. And so again, what was important to identify is just really to understand some of the, again, mechanics there of that blinking eye, understanding that in the center, if you see here with some of the modeling, and this is Hu and Vivek Shenoy here, looking at how pressure changes in that center region. You also get a fair amount of deflection in that center region. And it does impact the differentiation of these cells. So to maintain that epithelial phenotype of the cornea, you need that type of shear stress. And this is being used then to look at a variety of conditions, including the dry eye disease, and you can use this system to develop various therapeutics to identify treatments for those types of conditions. All right, and then I'm gonna walk through this, and because I've talked about a fair amount of work in the animal world. I'm going to finish out, then talk a little bit about some of the work that we've been doing in the plant world. And this really came about through, again, lots of effort here in the center, bringing groups together that were not talking to each other before the center, which includes myself, and working with plant biologists. I'm more of a traditional tissue engineer biomaterials person. And so it's my group, as well as Rom Dixon at WashU, and then Derek Dean at Alabama State, and then Marcus Faustin, who's also at WashU. And our challenge was, let's create a plant on a chip. Okay, we've got all these neat animal, devices. Let's do a plant on a chip. And why that's important, again, you want to look at, changes again at when cells assemble, but also you want to be able to probe different things similar to what you can do, get the cellular level and nuclear level with these plant cells. But in order to create that plant on a chip, well, how are you going to put those cells into a chip device, right? Was the main problem, or the main thing was, where do we start? So it was developing a scaffold for which these cells could attach to. And that was quite interesting. And so, again, depending on the plant cells that are used, in this case, we were using these tobacco BY2 cells. They're normally grown in suspension cultures. You maybe can put some things in agar, but generally they're in suspension cultures. So we threw kind of a lot of different biomaterials at these cells to see what would potentially work. And going back into some of my own work, we work with these piezoelectric biomaterials. So these are, again, a small deformation or mechanical force or deformation is applied to these materials, and you get electrical charge. And come to find out, again, your plant cell wall is made of cellulose, highly organized cellulose fibers. Cellulose fibers also have a piezoelectric property. So we tried those materials out, and lucky, we were able to get good cell attachment of those tobacco BY2 cells. And so we were able to grow those cells. They have similar morphology, the cells that would be grown in these kind of liquid environments. And what we were very interesting to find out was that they were attaching to these scaffolds via pectin. And so we're doing further studies to examine that, but that was also a quite interesting finding. So what about these piezoelectric fibers? Our understanding, we're still beginning to learn more about it, but in piezoelectric materials, you generally have these dipole orientations. And so what we found was that on the poled, kind of these negative side of these materials, that's where you're getting good adhesion of those cells, and to a lesser extent on the positively charged side. We also have been able to establish not only those tobacco BY2 cells can attach, we've just published there also that we can grow moss on these fibrous matrices. So now where are we with the creation of this plant on a chip? So we've been building, again, these PDMS devices, and we've now been able to put these scaffolds as well as those tobacco BY2 cells into those devices, and have applied some fluid. We've yet to construct a full device, but this is some promising work. At least we've been able to make a start here. So again, the idea here is to be able to apply certain forces, compression, tension, shear to these cells, and again, to mimic really what may be occurring either during development, or maybe it's due to wind or insects, what those types of forces that may be applied. And then really try to understand what's the response to those forces. And so shown here is, again, we can use a variety of techniques, including looking at calcium dynamics. Also, this is created also by the Dixon group and others. Okay, that's a little bit delay, seen in calcium sensor. All right, so a lot of these technologies then are actually making their way out there. So not only in use in the center, as I said, also in the scientific community, but we've also translated these into creation of these startup companies. So we have 9 startup companies created through the center, as shown here, many of which are doing very well with receiving venture capital funding. And there's many more technologies in the pipeline. All right, so I think you can, again, appreciate through the talks that you've heard yesterday and then today, how all these IRTs are kind of developing various either fundamental concepts or ideas, but now we're moving towards this integration of developing these technologies and understand this overall mechano-intelligence in these systems. And so again, important for fundamental understandings, but also again, technological innovation in both, right? Health, human health, as well as agriculture. So I thank you and hope to answer any questions as well. What we got? We're out of time.
We'll take 2 quick questions.
Hi, Treena. Thank you. This was really interesting. And I know you've covered a lot of people's work, but
Yeah.
question about the octopus structures. What about the shape of those structures evokes those different types of cellular differentiation responses? Like, what's the secret there?
That's a good question. And I wish Dan was here to answer that. But I don't know, Vivek, go ahead, let Vivek do that, yeah.
It's the anisotropy. So when you're holding it at 2 ends, your stress is anisotropic. That feeds back, aligns.
Oh,
So it has to do with if it's isotropic, then you don't, it's anisotropic feedback, that mechanical feedback, yes.
are you talking about his octopus device? Yeah, okay. Read his paper, I'm sorry. I wish I had the answer on that. Sorry about that.
Any other questions?